Hey everybody, Mr. Murphy here, and we are going to take a look at Chapter 4, all about cells. The chapter opens with this question about what do the characteristics of modern cells indicate about how cells first originated. This is a really good evolution kind of question where we want to be thinking about how the things that we can see and understand about cells today, not just in ourselves, but across all types of life, what that says about what our long ago common ancestor back at the beginning of life, what that must have been, what sort of components are essential to all life, and how could evolution from then till now drive the creation of these different mechanisms that we rely upon today. So important to just sort of keep that in the back of your head, and we'll see, I think, a lot of crossover between different cellular components, between the simplest prokaryotes and the much more complex eukaryotes, and it kind of becomes clear over time what the foundational needs are for something to truly be a cell. In an earlier class, I hope you've heard of cell theory. There's three basic tenets. So Robert Hooke was the first one in the 1600s to look at cells, and over the next couple of centuries, there was Matthias Schleiden, Theodor Schwann, and Rudolf Virchow, who really advanced that further in terms of a scientific approach. As microscopy became more of a available technology and a refined one, it allowed us to look deeper into what these components were that actually made up living things. And the three tenets of this are that cells are the fundamental unit of life, all organisms are composed of cells, and all cells come from pre-existing cells. I think it's important to pause for a second and just acknowledge that today this is not really fully embraced anymore, and a big reason for that is the existence of viruses. There is not a true consensus in the scientific community about an actual definition of life, and that can kind of seem surprising, but at the end of the day, we should remember that these are all just words invented by humans to describe things to other humans. So it doesn't really matter if something is alive or not alive, or I call it alive, but you don't call it alive. At the end of the day, there's much more to that conversation than we sometimes give it credit for. And that really is the nature of science, right? That this is not something where a textbook is written and we just believe it forever. A textbook is written as a record of sort of where we are so far. And then as new technology and better understandings come to light, the whole understanding evolves. So that's cell theory, which then supports this idea that, hey, if we're studying cell biology, that's really the same as studying life. So when we look at all these tiny microorganisms and single-celled organisms, that's okay. We're still studying life just the same as you or I. Well, not exactly the same, but that's sort of the point, is understanding what is it that does make something alive. The second point here, that life is continuous. Keep that in mind. Again, the theme of evolution weighs heavy over this entire course, and life goes back 3.5 billion years. And congratulations, you and I are among the survivors of that long history of living things, which is pretty cool. But it's kind of an interesting idea to consider the fact that every cell that exists in the world today has come from prior cells and prior cells and prior cells back to the beginning. And that'll be early life theories is something that we will explore later in this class. So a few fundamental things for us to really understand about cells. First off, cells are tiny. Almost every cell is tiny. There are some exceptions and fun little factoids about the largest cell ever and things along that line. But cells are really small, and the reason is this idea of surface area to volume ratio. Another idea that you've hopefully heard of, or at least one that we can kind of understand on a basic level, and we'll explore more in the course. But the idea here is that the surface area, that is like the outside area of a cell, has a certain value. There's a certain amount of surface area depending on the shape of a cell. But as things grow larger, because surface area is a squared value of, for instance, like the surface area of a cube is going to be six sides multiplied by the length times the width. whereas the volume of that cube is going to be the length times the width times the depth. And so it's a square to cubic relationship. And what that means on like a more easy to understand level is that as something grows larger, the volume is outpacing the surface area, which means that the contents, the three-dimensional volume inside of that cell, is growing faster than the surface area on the outside, which is to say that as a cell gets larger and larger and larger, its volume increases, its surface area will also increase, but at a slower rate. And that becomes a problem in terms of delivering the things that that cell needs across that membrane and being able to distribute it within. So a big idea in terms of helping us to understand why certain cells are shaped the way they are, but also why cells tend to be tiny. So, and just filling in what the slide says here, right? That volume, because there's more stuff, there's more metabolic activity, more chemistry going on, more needs, bigger demand, and that surface area is the gateway to getting things in or out of that cell. In this section, they provide this diagram. I think it's good to just sort of pause and examine this and understand just how small the things are that we are talking about. Because we exist in a much more macro world where we deal with things on like meter scales or maybe all the way up to like kilometer scales. If you look at a meter stick, the smallest little ticky marks on it are millimeters, which is, a 1,000th of a meter. It's still really tiny, but that's massive compared to the components of a cell and even the cells themselves. And so looking at this, you can see like a phospholipid, right? The component of the cell membrane is here, and that's somewhere in like a handful of 2 or 3 nanometers. A nanometer is a billionth of a meter. So we're talking about something that's a billion times smaller than the meter scale and still a million times smaller than like the smallest things that we tend to ever look at or measure. So we are talking about absolutely tiny, tiny things, which sort of explains why it took so much of human history for people to even become aware of these things, because we simply didn't have the technology to even allow us to observe it. So when we're looking at things under a microscope, for instance, plant cells, animal cells, things like that, they exist in this 10 to 100 micrometer or micrometer scale, so millions of times smaller than the meter scale. And that's sort of the limit of the classroom microscope technology that we have. Light microscopes don't go really beyond that too much. You'd have trouble seeing individual bacteria cells of most bacterial types on a light microscope, just because they are so small. You can see big clusters of them, and you can still gain some interesting insights. But really, the bulk of our understanding of all of these smaller things is only come in the past 100 years because we need better technology, things like electron microscopes, in order to visualize and understand how the structures of these things, what those structures are and how they influence what they're able to do. The last part of this section is just a reintroduction to the idea of the plasma membrane. Right off the bat, I just want to say plasma membrane, cell membrane, same thing. We're talking about the same thing. Don't get confused. Why do we use two different terms? I don't know. There's probably a reason. But when we talk about this, it's all the same. We're talking about the outside of cells. And there's some important roles. First and foremost, it is a barrier, and it is selectively permeable, which means that it allows some things in, but not other things. Also, it's important for communication. Cells, when they bump into each other, they don't have little eyes. They can't see each other, and so they see each other through interactions between the outside membrane layers of both of those cells. So there's going to be some important components that we need to understand that facilitate that. And then the last one, they need to be able to bind and hold together. The human body's, the adult human is about 35 trillion cells. It's not just a mountain of cells. We're not just like this gelatinous pile of cells just rolling around. It's held together, right? And our cells, therefore, need to have components to their cell membrane that help to produce that structure. So when we say this about cells, phospholipids and cell membranes and all of that, That is true for both prokaryotic and eukaryotic organisms. And in the next two sections, we'll go into what is a prokaryote versus a eukaryote. But very important up front is understanding the components that are the same between the two. And the two big ones featured here are that both are containing DNA. They contain the exact same type of genetic material, information material, and then the cell membrane. So the cell membrane between a prokaryote and a eukaryote, both are made out of phospholipids. And I included this diagram. It's not actually in your textbook, but the phospholipids are explained in a bunch of different places throughout the book, and this seemed like a good place just to remind you of what we're talking about. When we say phospholipid bilayer, what we're talking about is two layers, bilayer, of phospholipids. And phospholipids are these little guys here. It's the little circle with the two tails that we draw, and that is it's a triglyceride. It has two fatty acid tails, and the third group attached to the glycerol is this hydrophilic head, which has a phosphate group associated with it, and it is polar. And so that polarity is going to be important for us to remember and understand why it comes together the way it does, in that these fatty acid tails, which are very nonpolar, you notice that the two sides of this are mirrored, that the tails are all interior to the membrane, and that's because those nonpolar tails are all going to be commingled together because they are all nonpolar, and that then produces these two polar ends that are the hydrophilic heads, and those are going to exist a little bit better within the extracellular aqueous or watery environment and the intracellular watery environment. So when we say that these cells, or all of these cells, are made out of phospholipids, there's other stuff too within the cell membrane, but it is really a big ocean of phospholipids with proteins and carbohydrates and other things sort of peppered throughout. So in order to do these various things, really none of them would be possible if it was simply phospholipids, because phospholipids have a singular sort of chemical existence. So while they may be selectively permeable in that some things can pass through a phospholipid membrane and some things can't, it's important that when we think about the cell membrane as being selectively permeable, we understand that it's not just selectively permeable like in a static way, but that it's selectively permeable depending upon what it needs or what the internal or external chemical situation is. So, for instance, when the cell needs glucose, it can start taking in glucose. But there's other times when the cell may not be taking in glucose because it doesn't need glucose, or replace glucose with any number of different ions or molecules. So this is that's the beginning of chapter 4, just setting us up with a little bit of understanding about cells and the sort of universal components of them. And in the next section, we'll take a look at prokaryotes.